My name is Zhaonan from Kuala University. And in the recent decade, our team has developed an integrative ChIP-Seq database, ChIP-Atlas. And today I'd like to present you the overview of the ChIP-Atlas and then demonstrate its application, especially on analyzing drug modes of action and environmental pollutant health impact. And ChIP-Atlas comprehensively collected all ChIP-Seq data reported in research papers. And it provides a visual representation of transcription factor bindings to specific genomic regions or genes. And to create ChIP-Atlas, we initially downloaded all raw sequence data from ChIP-Seq experiments in public repositories such as in CBI and DDBJ. We then aligned all raw sequence to the reference genome, and then performed peak calling to identify statistically significant transcription factor binding sites. And all the results of this analysis are available on our website for public viewing. And in addition to this analysis, we also curated sample metadata for which we carefully documented hundreds of thousands of experimental data points to identify which transcription factor are investigated in which cell types. And through this data integration, in addition to simply viewing the experiment results, ChIP-Atlas also provides capability for comprehensive data mining. And this is the homepage of ChIP-Atlas. You can see a range of analysis tools for data mining. And as an illustration, the peak browser tool enables users to navigate peak call data from ChIP-Seq experiments. And by selecting genome assembly and cell types, and then simply pressing this button, all relevant ChIP-Seq datasets should be automatically loaded into Genome Browser, IGV. And then users can easily browse how different TFs bind to the genome. And ChIP-Atlas currently contains hundreds of thousands of ChIP-Seq datasets with an additional 60,000 of bisulfite-seq and 80,000 of ATAC-seq datasets added in an update 2 years ago. And of course, bisulfite-seq and ATAC-seq data can also be easily viewed in IGV through using peak browser tool, and in the same way as ChIP-Seq dataset. And another valuable tool provided by ChIP-Atlas is called enrichment analysis, which makes full use of hundreds of thousands of ChIP-Seq datasets to help users to identify transcription factors enriched to multiple genomic regions or genes. And here I would like to provide you a practical example on how enrichment analysis is used. And suppose this is our genome. And in recent years, genome-wide association studies have identified a huge number of SNPs associated with thousands of diseases and phenotypes. However, since most of these SNPs are actually located in non-coding regions, such as enhancers, it is very difficult for us to understand how these SNPs actually affect gene expression and contribute to the onset of diseases. Here we suppose that the TFs that commonly binds to, in another word, enriched to the SNP regions might be the candidate key factors involved in the onset mechanism of the diseases. And enrichment analysis is exactly useful for us to identify TFs enriched to a set of disease-associated SNPs. And by selecting genome assembly and cell types, users can then input hundreds of genomic coordinates of SNPs in this text box. As an example, we input hundreds of SNPs associated with atrial fibrillation, and clicking this submit button, this tool will then return results that indicate that estrogen-related receptor gamma was the top enriched TF around the input genes, around the input SNPs. And to confirm whether and how this ERG is involved in the onset mechanism of atrial fibrillation, we then conducted inhibition experiments on ERG using cardiomyocytes derived from human iPS cells. And the results indicated that ERG inhibition results in irregular motion of cardiomyocytes, which is quite similar to the phenotype seen in atrial fibrillation patients. And these results suggest the effectiveness of enrichment analysis in analyzing key factor, a potential key factor involved in the onset mechanism of genetic diseases. And enrichment analysis accepts not only genomic coordinates, but also gene symbols as input. And if you have conducted an RNA-seq experiment and identified a bunch of differentially expressed genes, you might be interested in understanding their upstream regulatory mechanism. And this enrichment analysis tool enables users to identify TFs that commonly binds to the input genes, therefore enabling them to identify the master regulators which co-restrict their expression. And as an example, we input a set of DEGs induced by drug treatment. And this tool will enable us to find TFs enriched to the promoters of these DEGs, and these TFs should be TFs that plays pivotal roles in the drug modes of action. And this tool will also assist us to predict potential applicable diseases of the drug. And as an illustration, by entering hundreds of DEGs induced by valproic acid, which is known as an convulsant, and also known as increase the risk of autism spectrum disorder in neonates when administered to pregnant women. And we found that CHD8, which was previously reported as a causative factor of ASD, was the top enriched TF around the input DEGs. And this suggests that VPA may contribute to ASD through the inhibition of CHD8 binding. And in summary, this enrichment analysis tool will help assist users exploring TFs enriched to drug-induced DEGs, and therefore helps them to predict potential disease applicability and side effects of drugs. And the main feature of this methodology is that it is totally independent from chemical structure of the input drug, which makes it advantage for analyzing complex mixtures such as traditional herb medicines. And we actually used this methodology to investigate mechanisms through which environmental pollutants may harm our health. And due to the time limitation, I cannot present the detailed results here. So if you are interested, please stop by my poster. And I have presented several case studies illustrating how this ChIP Atlas database can be used. So in a word, there are many ways to use it, and I will be happy if it can accelerate your research. And in closing, I would like to express gratitude to those who has supported my research. And thank you very much for listening.
Variable result, I think, but depending on the cell, the pH binding pattern is totally different, I think. So how can I overcome such limitation through the bioinformatics technique or something? Or we have to make much data, or so do you have any ideas for overcome such limitations?
I think I think there are many missing values that which means that there are many cell types that no ChIP-Seq experiments were performed. So to use, for example, machine learning to imply these missing values I think is good, but however, it's not actual experiments. So how to use these implied results is difficult.
Yeah, I agree. So, thank you. So, do you have any comments or questions?